Imagine a battlefield where the front line is not a trench, but the microscopic architecture of a living cell. It is a blunt instrument, a chemical storm that ravages the healthy alongside the malignant. But what if we could trade that storm for a surgical strike? Research shows we are no longer confined to the realm of science fiction. In a recent, landmark study that has sent shockwaves through the oncology community, engineers have developed a fleet of autonomous nanobots that have achieved a 100% success rate in mouse trials. No recurrence. No systemic toxicity. We are standing at a precipice. The data suggests that the era of flooding the human body with toxic compounds may finally be reaching its sunset. Why does this matter right now? Because cancer remains one of the most formidable adversaries in human history, claiming millions of lives annually while we rely on methods developed in the shadow of the 20th century. In this video, we will pull back the curtain on this microscopic revolution. We will examine the mechanics of these programmable nanostructures, analyze the data behind their flawless performance in the lab, and investigate the monumental challenges that lie between this breakthrough and the bedside. The trajectory of medicine is about to pivot. Let's look at how these nanobots navigate the treacherous geography of the human body to find, target, and dismantle the very foundations of disease. For decades, the battlefield of oncology was defined by a brutal, blunt force philosophy. Surgery was our primary scalpel, a physical excavation of the body to excise tumors, yet it remained forever haunted by the risk of microscopic remnants, rogue cells left behind in the shadows of healthy tissue. It was a necessary evil, a scorched earth campaign that flooded the bloodstream with toxins. We poisoned the patient to starve the parasite, praying the host would endure the fallout longer than the malignancy itself. The limitations were stark. This lack of specificity created a ceiling on efficacy. We were limited by the toxicity thresholds of the human body, forced to pull back the treatment just as it began to gain ground. The holy grail of oncology wasn't a stronger poison, but a smarter courier. This dream birthed the field of nanomedicine, the transition from systemic bombardment to molecular intelligence. By shrinking our tools to the scale of viruses and proteins, we moved the theater of war from the entire body to the precise coordinates of the disease. These synthetic architects could be programmed to ignore the healthy landscape, traversing the complex highway of the circulatory system without triggering a massive inflammatory response. We stopped trying to drown the cancer and started learning how to dismantle it from the inside out. This isn't just a refinement of existing tools, it is a fundamental rewrite of the medical operating system. But the true breakthrough wasn't just in the design of these machines, it was in how they learned to communicate with the environment of the tumor itself. That process begins with an unlikely fusion of biology and engineering. Imagine a submarine the size of a single protein molecule, drifting through the high-velocity currents of an artery. This is the realm of the nanobot. They are built from synthetic DNA origami, folded into complex, geometric cages that remain inert as they navigate the arterial highway. They are programmed with a biological compass. Our nanobots are equipped with molecular logic gates. These are tiny, chemical switches that only flip to the on position when they detect the specific, jagged protein signatures unique to malignant cells. Think of it as a biological keyhole. The robot remains locked, harmless, and silent until it physically docks onto the surface of the enemy. Once the connection is established, the machine undergoes a structural metamorphosis. It is a calculated collapse. As the cage opens, it releases a high-density payload of thrombin, a powerful clotting agent, directly into the tumor's blood supply. The malignancy is effectively quarantined, starved of oxygen and nutrients. Because the robot is blind to anything that doesn't share that specific, diseased protein signature, the surrounding healthy cells remain entirely untouched. They don't even know the battle is happening. It is a feat of engineering that turns the tumor's own hostile environment against it. But while we have mastered the art of the blockade, the question remains, what happens once the tumor is starved? The answer lies in the most complex weapon we've ever deployed, the nanobot's ability to recruit the host's own immune system into the fray. Imagine walking into a clinic for a procedure that feels less like a battle and more like a routine checkup. This isn't science fiction, it is the inevitable horizon opened by this breakthrough. Traditional chemotherapy acts like a wildfire to clear a single patch of weeds, scorching the landscape to reach the target. They do not poison the patient, they dismantle the disease. Patients won't be fighting to regain their strength after treatment, they will be maintaining their vigor throughout it. The economic implications are equally seismic. If we move from a model of long-term institutional reliance to one of rapid, outpatient precision, we unlock an unprecedented redistribution of resources. This is a transition from reactive, high-cost crisis management to proactive, streamlined intervention. The leap from the mouse model to the human trial is the final bridge we have to cross, but the blueprints are already drawn. We are moving toward an era where the word diagnosis no longer carries the weight of a life sentence, but rather the temporary inconvenience of a brief, elegant solution. The wreckage of the tumor is being cleared. The era of the scorched earth policy in oncology is drawing to a close, replaced by a sophisticated, quiet, and absolute victory. It is found in the invisible. If you want to stay at the forefront of this silent revolution, subscribe and hit the notification bell. The countdown to a cancer-free world has officially begun.